So one of the important aspects of the paper is that the inducer, retinoic acid, induces the expression of a gene called HOXB1. And the way in which retinoic acid works is by binding to the receptor, the RXR-RAR retinoic acid receptor. And this retinoic acid receptor is itself a transcription factor that binds to a very specific sequence of DNA called a RARE. Okay, and here you see the AGGTCA that's bound by the retinoic acid receptor. Now, that retinoic acid receptor not only has a DNA binding domain that allows it to bind DNA, a ligand binding domain that allows it to bind to retinoic acid, but it also has an activation domain that allows it to bind to some other protein. Now, when that receptor is not bound to retinoic acid, it binds the PRC2 complex, which then inhibits transcription of HOXB1. And when retinoic acid is bound to the receptor, it binds to the trithorax complex, which then, according to the model of the paper, induces HOXB1 expression. So one of the important things that the paper does is that it verifies this. It verifies that in the presence of retinoic acid, HOXB1 is expressed. Let's take a look at the data that demonstrates that. So what they've done is they've taken NT2D1 cells. Now these are human embryonal carcinoma cell lines. So it's a model system for looking at human cells. And what they did was they treated the cells for 24 hours with retinoic acid. And then they measured the amount of messenger HOXB1 messenger RNA using, they say here qPCR, but since it's messenger RNA, it must have been RT-qPCR, reverse transcriptase quantitative PCR. And here the minus means no retin, the control cells that have not been incubated in retinoic acid. And then the plus are the cells, the experimental cells that have been incubated in retinoic acid. And you can see that it's pretty clear retinoic acid induces the expression of HOXB1. Good. So, verified. Now, the question is how? What are the mechanisms by which retinoic acid is causing the expression of HOXB1? Basically, they want to know, is JMJD3 and UTX involved in this retinoic acid-induced HOXB1 expression? Is the trithorax complex involved? Is it necessary, right? They want to start to see what is the mechanism by which HOXB1 expression is induced by retinoic acid. So the first thing they do is what other changes occur in the presence of retinoic acid. So let's take a look at that experiment. So what they've done is a chip experiment. Now, we can ignore this. This is that same figure that we just looked at, okay? This is not anything this is a completely different experiment than these four panels here, right? This is measuring the HOXB1 messenger RNA in the presence and absence of retinoic acid. But let's focus over here. These last four. What these last four represent are chip using NT2D1 cells. Okay, those are the same type of cells. And the cells were untreated and treated with retinoic acid. So this is a chip experiment, chromatin immunoprecipitation, in which what they're doing is they're taking a whole bunch of cells. Some have been incubated in retinoic acid, and another set has not been incubated in retinoic acid. That's the minus plus here again. And then they took those cells, they performed CHIP, and they enriched the DNA, okay, with antibodies that bind to UTX, H3K27ME3, so the specifically trimethylated version of K27 of the histone H3 protein. EZH2, this is a component of PRC2, and a specifically modified histone H3 that is trimethylated at K4. So they wanted to see, how many of these copies of DNA, this DNA right upstream of HOXB1, right around the promoter region, in fact, probably right where a rare sits, right, that retinoic acid response element. They wanted to know how many copies of that DNA in these cells are bound by these various proteins. Now, what you see here is when they use the negative control, hemagglutinin, which does not even exist within the cells, good, you get almost nothing. Nothing, nothing, nothing. So those controls are fantastic. And then, when you take a look at UTX, basically how many copies of this portion of DNA are being bound by UTX, what you find is that only in the presence of retinoic acid do you get lots of UTX bound to this, or lots of this DNA bound by UTX. Look at this. Now UTX, remember, is sort of a proxy for trithorax. EZH2 is kind of a proxy. It's a component of PRC2. Look at this. Only in the absence of retinoic acid do you have lots of that DNA bound by EZH2. Okay, let's see what else. Only in the absence of retinoic acid do you have lots of trimethylation of K27. Look at this. The trimethylation of K27 is opposite of UTX. Well, that's beginning to make sense. It doesn't prove anything yet, but it sure makes sense that the more UTX you have at this position, the less trimethylation you have, because remember, the proposed function of UTX is a demethylase. And then, again, look at this. Only in the presence of retinoic acid do we have lots of trimethylated K4. Okay, so let's put this together into a model. What they've just found in this experiment is when you add retinoic acid to the cells, you get HOXB1 expression. Good, we know that happens, and that was with the previous experiment. We know that now from that other experiment we just looked at, when you add retinoic acid, the trimethylation of K27 goes down and the trimethylation of K4 goes up. When you add retinoic acid, right, the amount of K27, so the demethylase, so UTX, right, that sits at that promoter sequence goes up. And finally, when you add retinoic acid to these cells, the amount of this EZH2 protein, this is the K27 methyltransferase, right? This is the thing that actually adds methyl groups to K27. That goes down in the presence of retinoic acid. Okay, so now we have all of these correlations. And when you add retinoic acid, you get this event occurring, you get this event occurring, you get this event occurring, you get this event occurring. Now, the question is, does retinoic acid cause each one of these events in parallel, like what's shown here, that each event is separate from the other? Or, and this is what the authors are suggesting here, that it is sequential, that retinoic acid causes UTX to bind to the retinoic acid receptor, which then causes the demethylation of K27, which then causes activators to bind to the promoter of HOXB1, which then causes HOXB1 expression. This is what the authors suggest is happening. And so now we can start making some predictions based on this particular model. And the authors do this. They say, well, if it is sequential, then if we get rid of, or at least knock down, the amount of UTX within the cells, we should not get as much demethylation, right? The amount of demethylated K27 should go down. Right? We should have more trimethylated K27 because that's what UTX does. It demethylases K27, and if it's not around, we should have more than we should trimethylated K27. Okay, so, and that's only going to happen if retinoic acid works through to bring K27 in, which then causes the demethylation of K27. So let's say if that's true. So what they've done then is this experiment here. They have taken and used a chip experiment using NT2D1 cells again. This time, the cells have been transfected with either short hairpin UTX or short hairpin scrambled. Now that scrambled is going to be our negative control, and the UTX, that's going to be our experimental. Let's see how they did this experiment, because this is perhaps one of the most confusing ones. Here are the two cell lines they used. This cell line here is the control, the negative control, in which they transfected the cell with a vector that expresses a short hairpin microRNA that targets the scrambled messenger RNA. The only thing important here is that scrambled microRNA is not going to target UTX messenger RNA, which means in these cells, the UTX protein is going to be just fine. And then they did another cell type, the experimental, in which they transfected it with a vector that controls the short hairpin UTX. This is an expression vector that will express a UTX microRNA that binds to UTX messenger RNA and knocks it down, and therefore knocks down UTX protein. So there's less UTX protein in these cells here. Finally, each of the two cell types were were incubated with retinoic acid, okay? Which means that what we have going on, okay, is retinoic acid, according to the model, should be bringing the UTX to the promoter region, which should be demethylating K27, which should be bringing in the activators, which should be giving us HOXB1 expression. Okay, so that's what should be happening. Now we do a chip on these two cell types. First, we do our control with HA, right? That's our negative control. We shouldn't see any HA accumulating at the promoter region of HOXB1, and sure enough, regardless of whether we have cell type A or cell type B, we don't see any. And regardless of whether we have it in the negative control, right, with the scrambled, or the experimental with the UTX, doesn't matter. All right. Then we do another control. We want to make sure that the amount of histone H3 that is wrapped up in nucleosomes in this region is constant, that's not what's changing. And sure enough, you see, regardless of whether we're looking at cell type A or cell type B, the amount of histone H3 that is bound to this region here is consistent, is constant. That is not changing. So here's the big question. Our prediction is that when you take away UTX, you'll have more trimethylated K27 than you normally would. All right, let's see. That's over here, our chip experiment with K27. Cell type A, that's with UTX and the UTX protein intact. That's our negative control. And sure enough, you see a little bit less trimethylation, right? A little less of this K27ME3 at the promoter. And then when you take away UTX protein, boom, it shoots up. That is consistent with our prediction. You take away the UTX protein, and you get more K27 trimethylation. Why? Because UTX is what demethylates it. So if you take it away, there's nothing to demethylate it. All right, so far, so good. What seems to be happening is retinoic acid is inducing HOXB1 expression. Let's go back to this here. Inducing HOXB1 expression by recruiting the trithorax, including UTX, which then itself recruits our K4 methyltransferase. So K4 gets trimethylated, K27 gets demethylated. That's the epigenetic mark that attracts an activator, which then attracts the transcriptional complex, which turns on HOXB1 expression. And so we've gotten rid of UTX and showed that there is an increase in trimethylation of K27 when compared to a cell that does still have UTX. So the next question then is this. Now, let's get rid of UTX and see what effect that has on HOXB1 expression. Because if it's true, if retinoic acid works through UTX, right, that it's not independently turning HOXB1 on in parallel, it's working through UTX, then if we get rid of UTX, we should have less HOXB1 expression even in the presence of retinoic acid. Let's take a look at that experiment. So what we have here is a knockdown experiment, right? We've got those two cell types again. Our cell type that is our negative control that has the microRNA that makes scrambled. And then we have two constructs here, so two cell types that knock down UTX. And in this first one, we just support the idea that yes, the cells that express the short hairpin UTX1 or UTX2 do indeed knock down UTX messenger RNA. Okay, that's just the control. Here's the experimental. And look at this. We're looking at now what effect does knocking down UTX messenger RNA have on HOXB1 expression. Okay, we want to look at in the presence of retinoic acid because that's when we know we get normal induced HOXB1 expression that is induced by the presence of retinoic acid. So that's our positive control right there. Look at that. Wonderful. And now when you take away UTX protein, look at how much you get. You reduce that HOXB1 expression. Aha. So that's consistent with the hypothesis here that retinoic acid turns on HOXB1 expression through UTX. So that if you take away UTX, you get less expression. So, all together, what this data supports is the idea that the retinoic acid induced HOXB1 expression works through the trithorax complex, including UTX, which is the K27 demethylase, works through MLL, which is the K4 methyltransferase, which then leads to the expression of HOXB1. And that if you get rid of UTX, you get less HOXB1 expression. If you get rid of UTX, you get less demethylation of K27. Right? So they've supported some data that supports this particular model. And again, that goes on, and we can kind of look at it in a bigger picture, that this supports the model in which we have sitting at a rare upstream of HOXB1 gene itself, a retinoic acid receptor that sits at the rare site. If it doesn't bind retinoic acid, then the receptor is binding the PRC2 complex. If it is binding retinoic acid, then it binds to the trithorax complex, which then turns on HOXB1 in the presence of the PRC2 complex that represses HOXB1.